Let's Talk About Pain, a podcast from the Quebec Pain Research Network. In each episode, we discuss the latest advances in pain research with guests from across Quebec in the health and research communities. Enjoy your listening.
Hi, and welcome to this new episode of Let's Talk About Pain, a podcast created in collaboration with the Quebec Chronic Pain Network. For this episode, I'll be switching to English. My name is Carlos Gebers Montoro. I'm a postdoctoral fellow at McGill University under the supervision of Professor Mathieu Roy. This episode was recorded at the World Congress on Pain, held in Amsterdam in August in 2024. And our guest was Jamie Moffa, an MD PhD student at Washington University studying the neuroscience of opioid medications and addiction. Jamie is passionate about science communication and is the host of the podcast In Plain English. In this episode, Jamie shares their journey in pain research, the evolution of their work, and how their interest in science communication have shaped their career. It's a fascinating conversation, so without further ado, let's dive in. Here's Jamie Moffa. Hello, here with us, Jamie Moffa. How's it going?
It's going good. I think the jet lag is finally wearing off.
Yes, I think it will be completely gone when you have to go back, unfortunately.
That's true. Yeah, that's always the way it works. You're like, I'm finally adjusted, and now I have to return to a different time zone entirely.
Well, I really appreciate that you're taking the time to do this with us. It's really a pleasure to have you here. Just for the audience, I'm going to give a little bit of brief introduction about you, but then I would like to ask you also to please tell us a little bit more about yourself.
Yeah, for sure.
So we're here with Jamie Moffa, who's fellow Napster. We both did the North American Pain School this year, 2024, and it was a lot of fun. Jamie is currently an MD PhD student at Washington University, or WashU. I actually mixed both of them in St. Louis, United States.
Yes.
But, who is Jamie? Can we know a little bit more about you, just at the personal level, before we dive into the pain research?
Yeah, for sure. So I am actually originally from Connecticut. I grew up in northern Connecticut, went to university at the University of Connecticut studying neuroscience, and then decided to go to med school at Washington University for a bit of a change of pace. I actually didn't start out MD PhD, funnily enough. I started out as an MD student and then decided to transfer into the MD PhD combined program after my second year of med school because I missed research. I wanted more of what we're doing right now. So.
But does that mean that research was not your first choice for your career?
Yeah, I was initially thinking of just being a medical doctor.
What made you change? What happened? Or I'm aware of some of your previous research before pain, so maybe you can tell us a little bit more of what attracted you to research?
Yeah, so in undergrad, I had a couple of different research experiences, actually. All of them centered in the brainstem, which I still work in. But sort of my initial experiences were on brainstem regulation of breathing, which obviously ties into the opioid work that I'm doing right now, just slightly different areas of the brainstem. And then also I did a lot of in vitro kind of like plasmid design, very sort of molecular work as well. And interestingly, even though this was not the plan, my PhD has ended up kind of combining those two different areas of my background.
Wow, that's a beautiful, not ending, but it's a beautiful start actually to a promising career, I'm sure. So let's do some of this. Do you want to tell us a little bit more about your kids, the RVM, the medulla, the brainstem? What do you love about it? Why should people interested in pain or people living with pain know more about this?
Right. So the area I'm interested in is part of the very base of the brainstem. It's right above your spinal cord. And like Carlos said, it's called the RVM. And the reason why it's so important is it's kind of this hub for all of the signals that are coming from the brain that are actually controlling a bit of how you feel pain. A lot of people just think that pain is something that happens to your arms or your legs, somewhere in your body, and your brain just receives that signal. But your brain actually is able to kind of turn up or down the gain on that pain signal that you are getting from your body. And the RVM is kind of the last stop on the way to that, controlling the pain signal that's coming up from the periphery. So it's super important to understand how it's able to do that. And one of the ways that it's able to do that is by using opioids that your own body makes, and they bind to little proteins on the neuron surface that are called opioid receptors. And that's one of the key ways that this part of the brainstem is able to actually exert that control on pain.
Beautifully said. I've heard so many explanations about descending pain modulation, but this may be one of my favorites for sure. So
I appreciate that. Thank you.
Thank you for that. I don't know if you remember, we did this at NAPS. You had to explain your project in a minute, then in 30 seconds.
Yep.
So that was actually very good. We're not going to do that experiment right now.
Yeah.
But honestly, timing was perfect. So I appreciate that. And I think a lot of people will be interested in what you were talking about, the endogenous opioids, those endorphins that we all hear about. Is there a way, or do we know ways that our own brainstem is able to engage these endorphins or these tracks a little bit better? Are you working on that directly, or do you plan on working in translational aspects of your research?
So my project right now is very heavy in the preclinical foundational science. It's not directly clinically relevant right now, but I think that understanding how those endogenous opioids are able to exert control is really important. And actually, there's been a lot of other cool research here that's not necessarily my own that is showing some of the ways in which the opioid system is engaged in this top-down control of pain. One of those ways that I've learned about that's really interesting is that the opioid system in the brainstem is really, really important for the placebo effect. So when you think that you're getting something that's going to reduce your pain, even if it's just like a sugar pill or something that's totally inactive, one of the main ways that actually works, that your brain sort of tricks you into thinking that you're actually feeling less pain, is through the opioid system in the brainstem. So you actually can trick your brain into turning on by thinking that you're receiving something that's going to make you feel less pain.
Many people in our audience are going to be interested in hearing more about those molecules, the endogenous opioids or endorphins that we've all heard about so much. So I was wondering, since you're studying mostly in mice and you're doing what we call basic science, is there a way that we can measure these in animals, like these potential placebo mechanisms or placebo effects?
I went to a poster yesterday that was actually about the placebo effect in mice. And you might wonder, how can you trick mice into thinking that they're getting pain alleviation when they're not, because you can't tell them, this is this kind of drug. But what they did was actually they trained mice on a hot plate with two different temperatures on it.
Mm-hmm.
And they started out, they taught mice that, one side of the hot plate was hot and the other side was a neutral temperature. They taught mice they could escape from one side to the other.
Mm-hmm.
And then they put half of the mice on a hot plate that both sides were the same temperature.
Okay.
Both were a noxious temperature, so it would be hot to them. But the mice that had received the placebo training, that had learned that they could escape from one side to the other, did show a preference for escaping from the side they thought was hot to the side they thought was not hot, which is similar to the placebo. They thought that they were experiencing less pain on the side that was supposed to be not hot, even though they were the same.
So I guess this is attained through conditioning mechanisms, potentially?
So they're conditioned initially on one side's hot, one side's not hot, or the control group was conditioned on both sides were equally hot.
Okay.
And so only the animals that had been conditioned on the one side hot, one side not hot showed this preference when they were put onto the hot plate where both sides were the same.
So I guess that also means that these regions that then descend to the spinal cord and inhibit pain, they are controlled by higher cortical centers, so other regions of the brain. Are there specific regions you're interested in that do relate to this RVM region of yours that you really like to study?
Yeah, so the closest relay is another area of the brainstem. It's called the PAG, the periaqueductal gray, and I can kind of break down what that means. Anatomically, it means that this is a region of the brain that is composed of gray matter, which means cell bodies. So neurons have cell bodies and they have projections that go and contact other neurons. And gray matter is mostly the cell bodies and white matter is mostly the projections. So this is gray matter, and then it's around what's called the cerebral aqueduct. Your brain is bathed in this whole fluid that provides it with nutrients. It does oxygen exchange. It's like super important, and this fluid flows throughout the brain, and this aqueduct is one of the ways that the fluid flows. So you've got this brain fluid that's flowing through the middle of this group of cell bodies, and that's what the periaqueductal gray is.
Just a small addition, I've heard this fluid referred to as brain Gatorade, which I thought was brilliant. Yeah, because it doesn't only bathe it, right? It also provides, well, you did say, provides nutrients.
Nutrients, sugar, all that, yeah.
So this is also called cerebrospinal fluid. So the PAG is also one of your targets in your research. Are you also looking into it?
So part of my project did look into that, and so the PAG is also important in this descending pain control. It's also, people have done studies where they activate the PAG and then the mice feel less pain. So it does send those projections that are coming from the cell bodies to the RVM. So it's kind of the relay stop before the RVM that's participating in integrating a lot of the signals that are coming from higher brain regions. But even upstream of that, you get some parts of the brain are involved in sort of the more emotional aspect of pain. So not just that you feel pain, but that you feel bad about feeling pain.
Yes.
Some parts of the brain are more involved with the motor aspects of pain. So how you're going to react, how you're going to escape and avoid that pain. So once you get up above the spinal cord and brainstem, you get into actually a lot of very complex processing of how we feel pain, respond to pain. And a lot of that's going to be really important for treating pain, because pain's not just the sensation, it's also all of the other inhibition on quality of life and negative mood that come along with
So this makes a lot of sense to me now that you're saying that, although I've studied this before, but I realized that, for example, when you're playing an important game, you're an Olympian, you're an athlete, you could get injured, but maybe not feel the pain necessarily for a little while, because some parts of your brain are telling you that that's not important at this moment. So this is engaging these systems, sorry?
It is, for sure. And it's a phenomenon that researchers like to call stress-induced analgesia, meaning that when you're in a situation of high stress, you are less likely to feel pain that is happening, because I don't know, the one I like to use is say you're running away from a bear. It's not really all that important if you have a bunch of cuts on your leg, because you still need to escape the bear. But then after you're safe and you've escaped the bear,
that's when the pain hits.
That's when the pain hits. That's when all of those adrenaline and everything dies down, and then you're like, oh my God.
Luckily, we don't have to run away from bears too much anymore.
No.
At least not here in Amsterdam. This is where we are right now. But in Canada, this could be a thing. So
I was going to say where I'm from too. Northern Connecticut definitely has bears.
Yeah. So I just want to ask you a last question about your research, because you also mentioned opioids as medication, so exogenous opioids. How do people who take opioids, or how do their RVM change, or do they change? Have you investigated anything into this?
I have not.
Or is this something that's known, or is there an important fact that you would like to share about that?
In general, when you take kind of any drug that's going to mess with your central nervous system, your brain, how it's processing things, anytime that you take a drug that's going to modify how your brain works for a long period of time, your brain is actually very smart, and not just in the fact that all of our listeners know a lot of things, which I'm sure you all do, but your brain and your body want to achieve something called homeostasis. So it has this kind of set point that it wants to get back to. And flooding the system with exogenous opioids is taking the brain away from the set point that it wants to be at. In the first several days to weeks that you would be on one of these medications, it's going to be super effective, because the brain still hasn't figured out how to deal with that and bring itself back to that baseline that it wants to be at. But over time, it's actually going to remove some of those opioid receptors from the surfaces of cells and reorganize different circuits of connections and signaling inside of the brain to kind of mitigate the effect of that flood of opioids.
So to make itself a little bit less responsive, to respond less and have a more balanced response.
Yeah, so that's why those medications will tend to become less effective over time. So that's actually one of the reasons why opioids are not that good of a treatment for chronic pain, is because chronic pain is chronic, and so whatever treatment you have for it, you need to be on for a long time.
Yeah.
And if you're on opioids for a long time, the effectiveness is going to decrease. So opioids really great for like post-surgical pain. They're going to help out a lot with like the immediate aftermath of having your body cut into and done surgery on. But in the long term, one of the main goals of a lot of researchers here at IASP is about finding treatments for pain that are not based in opioids, because not only are they not that effective for chronic pain because of this, adjustment that your brain does, but also, as we all know, they're addictive and pretty dangerous in high quantities. So we want to find a drug that would be more tolerable at higher doses than opioids are.
I'm no expert on addiction, but I have a feeling that what you just explained could partially explain also the mechanisms of why we tend to get addicted to these substances, because we will need more doses to get the same result.
You need more doses, and in fact, so your brain's adaptation is kind of a double-edged sword, because not only do you need more of the drug to get the same result, but if you don't have the drug, you actually feel worse, because all of these receptors have been taken off the surface of your cells, and your brain has reorganized itself around expecting a certain amount of opioids coming in from the outside. And so if you don't have that anymore, you're actually going to feel pretty bad, because your brain is expecting this thing that it's not getting, and so you're going to kind of get the inverse of what you would feel when you're on opioids. So if you feel like sedate and like calm when you're on opioids for a long time, when you go off of them, you might feel agitated and like overactive, right? It kind of swings back in the opposite direction, which can also be dangerous if you are on a high dose of opioids for some kind of maintenance therapy. You want to go slowly so your body can readjust itself to having less opioids in its system.
Great, Jamie. Thank you for this overview of why everyone interested in pain research or living with pain should know a little bit more about the brainstem, the RVM, how the opioid system interacts in these regions. This has been great, but I know there's another side of you. Jamie is not just a great scientist, also a great person. I'm so happy and feel so honored to have met you at NAPS. It really brings me happiness to share these moments with you.
Thank you.
At the same time, Jamie is an amazing science communicator. So let's talk about Science in Plain English. What is that about?
In Plain English is my podcast. It's a science communication podcast that's all about talking about science in plain English. So for each episode, I bring on an expert in a field of research, and it doesn't need to be pain or neuroscience. I've brought on experts that are from immunology, like learning how your body responds to illnesses, or experts in things like anthropology and archaeology, learning about things that are totally outside of my wheelhouse and I never would have known about if I didn't do this podcast. So I bring on an expert, and I also bring on two people that know nothing about the subject. So the expert's going to talk about their field of research, present a paper using as little jargon as possible, and really walking the guests who are not experts through the paper. And then the guests ask the kinds of questions that anyone would ask when learning about that subject for the first time. We have really fantastic conversations. I think everybody learns a lot when I do these episodes.
Honestly, that's a great format. I had never heard it before talking to you. I don't know if you had an inspiration for it or you just came up with the idea, but congratulations either way.
Yeah, I'd been kind of thinking about it for a while, and what it came out of was I thought about the format of journal clubs, which in academic research are when you get a group of people together who all do the same kinds of research, and you present a paper, and then everybody talks about the paper. And it ends up being a really productive discussion about the science, and people come away with a lot of cool new ideas and knowing a little bit more about the field that they're doing research in. So I had this vague idea of doing a journal club, but for people who are not in science, because bringing this research to non-scientists is super important, because like in the U.S., at least, that's who funds it. If you pay taxes, part of your taxes goes to fund the NIH, which funds a whole lot of biomedical research.
Same thing in Canada. I hear you. Yes.
Yeah, and also it's like really relevant research, especially in medicine and like biology. It's very relevant research to people.
So at the end, they are the end users of this research, so they should be aware of what's happening.
Right, so they should know what's happening. And I felt that a lot of science communication efforts can tend to fall on the overly simplistic side, where like you can get a snippet of what's going on, but there's not actually a lot of detail there. So we take the time to dive into one paper per episode, and we're really like getting into how does this paper fit into the broader field of research, and like why was it important that these scientists did this paper, and what did they find?
And where can we find your podcast for our audience?
Yeah, so we have a website. It's called inplainenglishpod.org. You can also find In Plain English by searching In Plain English Science on any podcast platform that you like to use.
Perfect.
And we are on social media on Twitter and Instagram at Plain English Sci. That's SCI at the end.
Great. Thank you so much, Jamie. We're going to wrap up here. There's a lot of excitement building up, not just for this conversation, but also because we're going to soon be listening to two Nobel laureates that are going to be speaking for us at ISP. I'm sure you're very excited about that as well.
I am, yes.
So thank you so much for taking your time, and we really appreciate your contribution, and we will be looking forward to listen or to hear more about you, about your podcast, and about your research.
Yeah, thank you
Jamie,
for chatting with me.
Thank you. Our time with Jamie was cut short when the conference organizers called us into the room for the Nobel Prize winner's talk. That was a session we couldn't miss. We hope you enjoyed this conversation as much as we did. And from here, a big thank you to Jamie Moffa for sharing your story and passion. It was truly a pleasure having you on the podcast, and who knows, maybe next time we'll join you on In Plain English. And to all our listeners, thank you for tuning in to this episode of Parlons Douleur. Hasta la vista, à bientôt, and until next time, please take care.
Thank you for listening to Let's Talk About Pain. Don't hesitate to subscribe so you don't miss an episode, and to share the podcast with your friends. For more information, visit our website at qprn.ca or our YouTube channel by searching Quebec Pain Research Network. See you next time.